Hello everybody. I am Dr. Shabnam Das Kar, a specialist in functional and metabolic medicine. I'm in Alberta, Canada. And my guest today is Dr. Debojyoti Dhar. Debojyoti will introduce himself very soon. Today we are discussing a paper that Debojyoti has published recently. This is a review paper called Gut Microbiome and COVID-19, Possible Links and Implications. I'm looking forward to a great discussion ahead. And over to you, Debojyoti.
Thank you, Dr. Kar. It's nice to be back on this channel. My name is Debojyoti, and I'm the co-founder and director of Lucinrich Bio, which is South Asia's first microbiome company. And as I've told earlier, we have commercialized South Asia's first gut microbiome test called BugSpeaks. I have a PhD in molecular biology from Indian Institute of Science and a postdoctoral experience in UMass Medical School in the US. Post that, I've been part of many companies. I've looked at drug discovery, I've looked at corporate communications, business development, so on and so forth. And since for the last 3-4 years, we have been working on the microbiome aspect. I thought, why not, use that information knowledge to write a paper. And as we discuss the paper, I will tell you why and what cues I got to initiate that writing. So that's all from my end in the introduction part.
Thank you, Debojyoti. So are you going to share a PowerPoint?
Yes, I will.
Okay. Actually, okay, thank you. I can see your screen.
Okay. Let me go to the, is it visible?
Yes, it is. And I'm going to, I think I can, if I stop my video. Oh, sorry about that. Go ahead.
Okay. So as Dr. Kar during her introduction said, I've just published a mini review on COVID-19 and gut microbiota, possible role of gut microbiota in COVID-19 and its possible implications. This has got published in a peer-reviewed journal called Virus Research, which is online right now. You can click on the DOI mentioned in this slide and read the paper. And as I said, I'm the co-founder of Lucinrich Bio, and these are the sites of my company and the tests that we have launched. Okay, so let's go to the paper and some background of what we are discussing. So we all know right now, I think everyone is a coronavirus expert, which is a good thing. A lot of people have become immunologists, molecular biologists, and virologists. So just to give a small background, the coronaviruses basically are enveloped positive sense RNA viruses. Positive sense RNA viruses means, see, all these viruses have genetic material, either DNA or RNA. For the uninitiated, we know that DNA gets converted into RNA, and then RNA is the base for protein production. So positive sense RNA means the RNA which is there inside the virus is directly used for protein production. So this basically translates into protein without getting transcribed into another sense. That's why it's called positive sense RNA virus. And the name corona is because of the spikes on the surface, which gives it a crown kind of an appearance. Interestingly, the first coronavirus outbreak happened, the major outbreak happened in 2002-2003, which incidentally was called as SARS. The statistics here will show that it was more mortal than what we are seeing right now. In fact, it had a mortality rate of 11%, but it was contained pretty fast. Another bout of outbreak happened in 2012, and which was called as MERS, or Middle East Respiratory Syndrome coronavirus. As the name suggests, it happened in the Middle East primarily. And again, the fatality rate was pretty high and much higher than what we are seeing with the current coronavirus outbreak. So now this year, the last year, late last year, we had this coronavirus outbreak, which is now called as COVID-19, and it is caused by a virus now being rechristened as SARS-CoV-2. Why SARS-CoV-2? Because when the SARS happened in 2002, 2003, that was called as the SARS virus. And since it has 70% similarity with the SARS-CoV virus, it is called as SARS-CoV-2. Some of the clinical manifestations that we have been seeing on this particular disease, are fever, conjunctivitis, very interestingly, loss of smell and taste in some cases. In subset of patients, it progresses to pneumonia, failure, lung failure, and death. And this is mainly due to the extreme surge of inflammatory cytokines. So these are basically something called as cytokine storm that happens. And we'll discuss, it doesn't happen with everyone. It happens with, comorbid patients or people who are old and so on and so forth. Interestingly, there are a few gastrointestinal symptoms that is being seen in many of these patients. And that's where the thought of writing this paper actually came up. This is a virus which infects the lungs primarily, lower respiratory tract lungs, right? Then why is it that some of the people are getting gastrointestinal symptoms, especially diarrhea, vomiting, and nausea? So this was very interesting. However, this was reported earlier also. Anyway, so some of the facts that I've collated from some of the papers is that the median time for onset of symptoms to dyspnea is around 5 days. Progression to something called as acute respiratory distress syndrome or ARDS is 8 days in subset. Again, this is not for all. This is only for subset of patients. And complications include pneumonia, acute lung injury, even liver injury to some patients, and kidney injury. Very interestingly, this adverse outcome and death is more commonly seen in the elderly, immunocompromised patients and patients with other comorbidities. And for the people who have been following this channel, our first talk was on COVID-19 and gut microbiota, which Dr. Kerr had initiated. And there also, we were highlighting the fact that this is affecting more elderly and immunocompromised and patients with other comorbidities like hypertension and diabetes. And again, this also had given some food for thought to me to write this paper, because there are two things which are happening, and I'm now coming to the cues that that basically drove me to write this paper. The first cue, and as we were discussing in the previous slide, was the notable GI symptoms, right, which is abdominal pain, diarrhea in around 20 to 50% of the COVID-19 patients. Incidentally, Dr. Kerr, when the first reports came in of this link, let's say in March, late March, the percentage of patients, primarily that was coming from China, the percentage of patients with the GI symptoms was around 13%. But now, when you look at the reports coming from the US and even further reports from China, I think it has gone up to 60%. Mm-hmm. Again, that's the range, 20 to 50%. That's why I put this as a range. So preliminary findings published in a journal called Gastroenterology. Researchers examined clinical specimens from 73 hospitalized patients with SARS-CoV-2, and 39 patients tested positive. What is very stark here is that many patients, although they become negative in terms of their respiratory samples showing negative results, they are still positive in terms of their stool. That means the viral RNA can be still detected in the stool. What are the clinical implications for this? We don't know, but one possibility is always there, that this can have a fecal-oral route of transmission. No one has ruled that out, but I think it is also not yet confirmed. If I'm correct me if I'm.
So, Debojyoti, I'm just going to interrupt you there, and why both of us want to focus on the fecal-oral transmission is one of the things is the gut microbiome. And the other
Right.
thing is, in countries like India, we are still
Yes.
dealing with a lot of waterborne diseases.
Right.
So, as Debojyoti, you mentioned that we still don't know whether there's a fecal oral transmission, number one.
Yeah.
Number two, I think there was a paper which came out. So, they looked at the water in Schiphol airport and they
Yes. Yes.
found the virus. I think it was the virus particles, I'm not sure. But anyway, some virus.
RNA. They looked at the RNA.
Yeah, so they found some of that in the water there. Now,
Yes.
Schiphol is not a, I mean, that's not a country where they deal with,
Correct. Correct. Correct.
waterborne diseases. So the
Yeah.
other question I had, and again, I don't think we have the answers yet.
Yeah.
As you rightly mentioned, the nasopharynx was negative in many of these patients, but their anal swabs were positive. The
Yes.
question that we haven't been probably able to answer yet, and we'll probably have more data later on, is whether the positive anal swab is able to transmit the infection. Am I right?
Correct. Correct.
Yeah.
So that's a question that, in fact, there were few reports which suggest that they could. So there are two things, right? One is presence of RNA itself doesn't mean that you are a spreader, because the RNA can come from the virus getting lysed.
Mm-hmm.
The RNA itself is actually of very little information that in that manner, unless you look at the microscope and and look at the viruses. If the viruses are found in the sample, and I think there are few reports where they have also seen viruses to be present, not only the RNA, the viruses to be present, then it's a matter of concern that that it can be a source of transmission. But
Yes.
again, we are
Just one more thing to our listeners is, just because we don't have the information yet, it doesn't mean it cannot be transmitted. So I would suggest that the hand washing is extremely important. Another thing that I came across, and I did not delve deep, and I decided, okay, I don't need to know so much, I can just take the action. So this was about spreading of the virus while flushing a toilet. So do you know anything more about that? So what I heard is, when the toilet is flushed, a lot of it is spread in the aerosol. And one of the things that you mentioned, Debojyoti, is SARS, the old one, the 2003 one, had the mortality of 11%. MERS has a mortality of 34%. But COVID-19 doesn't have as high a mortality, but the spread has been much, much wider than the old SARS and the MERS.
Absolutely.
So would the viral load be one of the deciding factors? And that is why I'm bringing up the toilet seat thing. So this is, again, I'm ultra conservative. Maybe it's a good idea to put the lid down, when flushing.
Absolutely.
That should be the norm, but I think a lot of the time we don't think of it. So just one more thing, we don't know for sure. And the other thing is, if someone is self-isolating, probably wiping the toilet seat inside would also be one more factor. So these are just a few of my observations for people to implement for themselves. So carry on.
Absolutely. I mean, see, we are getting some clues. There are no definitive answers right now in terms of the transmission from the fecal route. But as Dr. Kerr is saying, we have to be cautious, because people are seeing viruses in the stool, and yes, definitely that makes sense, Dr. Kerr. Okay, now the third clue is that this virus basically attaches to a receptor called as angiotensin converting enzyme 2, or ACE2 receptor, which are found on the alveolar cells in the lungs. There are few reports and kind of confirmed reports where another role of another protein or a protease, which is TMPRSS2, has been found to be equally important. It is not only the attachment of the virus to the ACE2, but the serine protease, which cleaves a certain part of the the virus to make the attachment much more workable for the virus. So interestingly, ACE2 and TMPRSS2 are expressed highly in the GI tract. So no wonder that we see some of these virus particles in the GI tract. So And the final clue was, which we were mentioning in the earlier slide also, is that COVID-19 affects mostly the elderly, immunocompromised, and the comorbid patients group. Now, when you put all these points together in perspective, um, you would start to, question few things. One, why is this virus affecting only a subset of the population? Okay, there can be many reasons to it. Some of the reasons can be genetic. Some of the reasons can be based on molecular biology of how ACE2 receptors are being expressed on the cells. And Dr. Kerr, you would also we would have probably also looked at some of the reports where the role of progesterone has been shown.
Yeah.
Why males are affected more and females are affected less. So there are multiple reasons for this virus affecting a particular subset and not everyone equally. But one of the important subsets that may have missed or people are not looking into that in that detail, now things have changed, is the gut microbiota. And hence this paper to basically talk about that possible link and what can be the implications of such link if much further research needs to be done, but this is some very important part of the puzzle that needs to be looked into. So with that preface, let's get into the next part of the slide. One of the very important premise for the role of gut microbiota in lung infection, including COVID, is the fact that there is something called as a gut-lung axis. And if I remember correctly, we had dealt upon it a little bit on our first interaction. So basically, the gut is supposed to link with a lot of organs in the body, be it liver, be it skin, be it brain, be it lungs. And the gut-lung axis is very important here because we are dealing with a virus which primarily affects the lungs. So if I take you through the text here, basically this axis allows for the passage of endotoxin, microbial metabolites, cytokines, and hormones into the bloodstream, connecting the gut niche with that of the lung and vice versa. It's not only that the gut is affecting the lungs also affect the gut. Okay. Very interestingly, which I found, Dr. Kerr, is that structurally the respiratory and GI tracts have many similarities. Both extensive, highly vascularized luminal surface area, which is protected by a selective epithelial barrier and an overlying mucus gel layer from which protects them from commensal bacteria, pathogens, and foreign antigens. In fact, primarily they have come out from the same source of developmental biology.
Yes, Dr. Vijaya had talked about it at length. So we'll post a link to the previous video as well. That was a fascinating discussion we had.
Yes. So the links, therefore, the structural similarity therefore stems from that fact maybe. Now there are very anecdotal or lot of research has happened which has shown that infants or children who have dysbiosis in the gut, that means dysbiosis means the balance of good bad microbiome or microbiota is not correct. Many of them have been shown to have asthma in their later period of life. And there are numerous animal models that have shown the direct link of when you change the gut microbiota by giving antibiotics and stuff like that, it somehow affects the ability of the body to fight or ward off infections in the lung.
You're right, absolutely. And also one of the I think there are papers supporting the fact that children who are born by a cesarean section, they have a very different gut microbiota picture, and they have a higher risk of childhood asthma and allergies. So
absolutely,
definitely there is
there's a link, exactly. And what we are till now we were talking about the link, the gut microbiota to the lung, but there's a bidirectional link also. So it has been found that post sepsis, the lung microbiota shows presence of bacteria which originates from the gut. So that means there is something which is happening, which is making the microbiota in the intestine come to the lungs and populate it. Okay. Viral infection in urine models, that means the mice models, have shown decreased gut diversity and increased gut inflammation. And interestingly, and we have experienced this in humans also, when we have some infection, we generally report loss of appetite, right? We stop eating proper food and all. So that again, people have shown that it is somehow affecting the gut microbiota. It is leading to some TNF alpha going up and so on and so forth. So there are some evidences to suggest that there's a link between gut and the lung, and it's a bidirectional one.
Yeah, and Debojyoti, it reminds me of there is one saying, stuff a cold and starve a fever.
Yes,
So
yes, yes.
again, most colds are viral infections. So definitely
Yes.
this is probably ancient wisdom, and now we are having the scientific tools to, to be able to prove that. And when you talked about, malaise, body ache, not feeling well, so a lot of that is also the sickness behavior, right?
Correct,
So
correct.
sickness behavior, another thing that it would do is, again, this is again nature's way of doing a physical distancing. When someone
Yes.
is feeling sick, they would not
Correct.
go out into, into the public. Versus,
Absolutely.
so there are so many ways that nature has designed our bodies in such a way that it's amazing, really. Carry
Yes.
on.
Yes. Yes. And one of the things of what you mentioned about not feeling good, mentally feeling down, again there can be a gut microbiota connection. See, because your
Absolutely.
gut is getting affected because of the infection, and once the gut gets affected, as I was mentioning, the gut has links to the brain. There's a gut brain axis also, and in fact there's a theory of depression and anxiety linked to the
Yes.
gut microbiota.
Yeah. I think it's more of, it's probably becoming less of a theory, and we have probably the mechanistic connections as well now.
Yes, yes. There are
Yeah.
few studies and good studies.
Yeah, good study.
Anyways,
Yeah.
yeah, but and
Yeah.
the thing is, there are a few trials also which have taken place, which have tried to use probiotics to
Yes.
improve
So,
depression score
yeah.
and all.
And are you talking about that Emeron Mayer paper where they did functional MRI to women after they gave
I don't
them?
remember the
Yeah.
authors, but there are many actually.
Yeah. Yeah.
Um,
Anyway, let us get back to your paper now.
So, okay. So the next thing that that I discussed in the paper, the first one was, of course, the gut-lung axis. The second part was the role gut microbiota plays in immunity. Okay. Now, this is very important for us to understand because, you know, there can be two things. One is hyperimmune reaction, and the second one is, a hypoimmune reaction. So in both cases can lead to devastating effects in terms of especially in terms of COVID. So if you have a hyperimmune reaction, it will lead to the so-called cytokine storm, and then cause damage to the lungs, and then all those other things will happen. And if you have a hypoimmune reaction, again, then what will happen is the virus will take over the body in much faster manner than. So it has to be a homeostasis. The balance has to be maintained. And interestingly, gut microbiota uh, has a role in both innate and adaptive immune response. And there are reports which suggest that gut microbiota plays a role in the immune homeostasis as well. Without getting too much into the details of how these things happen, just this is a slide just to give you an impression of how these guys affect the various factors or effectors, as we call. So you have the T cells, the B cells, the macrophages. B cells, as you all know, produce antibodies. The T cells are there are various subsets of T cells now. In our days we only knew CD4 plus and CD8 plus cells. Now I think there are so many other subsets. You have Tregs, Th17 cells, and so on and so forth.
So Devajyoti, I'm interrupting you. Your days you knew that much. My days we didn't even know that much. So when I came across the dendritic cell, I
Yeah.
thought, what is the dendritic cell doing in immunology? Like that was supposed to be in the brain. So
Right.
my
Right.
knowledge of immunology had to be upgraded majorly, and I'm still not there yet. Anyway, thank you. Go ahead.
And it's an evolving science. I mean, as we move on, we will get to see many more subsets maybe. Uh, but yeah, I mean, see, the thing is, this is work in progress, right? I mean, there are so many things which are unknown, but whatever is known gives us a lot of clue or cues to what gut microbiota can and is doing to our health and wellness. So, so immunity, gut microbiota plays a humongous role in the immune system of the body, and I think our on the right side there's a box where I don't know whether people can see it because our videos are over the top there, but anyway, uh, it basically
Yeah.
gives an example of two bacteria. One is segmented filamentous bacteria, which leads to Th17 propagation, and Th17 is pro-inflammatory. And there are other bacteria like Clostridium species, which lead to Treg population going up, which is anti-inflammatory. So just to give you an idea of how the immune homeostasis can take place, which is probably dependent to some extent, on the gut microbiota, which led me to to think on these lines basically. So basically, what this slide shows, which is in the paper, is that you have the COVID-19 infecting the lungs. You have, if you have an overreaction, then that means the pro-inflammatory response like Th17 subset cells that we were discussing, IL-1, IL-6, TNF alpha going up, and you see the balance is tilted towards more of the hyperimmune reaction, which is leading to ARDS and so on and so forth. Whereas when you have underreactive immune response, again, this can also cause the COVID-19 to cause the disease. So the balance, therefore, is very important. And as I explained in the earlier slide, that gut microbiota might play a very important role in the balance of this particular thing. Now we come to the very important aspect, and I think both Dr. Kerr and I know that one slide or even 20 slides are not enough to talk about the role of diet and gut microbiota. There are so many things to really talk about on all these things. So without doing that in this particular session, uh, suffice to say that gut microbiota is malleable. We had touched upon this on our first session, and diet, as Dr. Kerr had also put forth on that occasion, that diet, lifestyle, your exercise, etc., medicines like antibiotics, and even your artificial sweeteners, all of them affect our gut microbiota. So 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 it is very important that we strengthen the reservoir, so to speak, and that's what I have tried to put in the paper. Just I'll just go through these texts and explain this. So apart from the intestinal disorders, which are directly linked to the intestinal microflora, for example, even diabetes, obesity, diabetes is also supposed to be linked, excuse me, linked to the gut. Uh, Colon cancer, inflammatory bowel disease, etc. Changes in dietary patterns and their effects on gut microbiota are implicated. So a lot of work has been done to show that how they are linked. Now, type of diet influences the kind of microbes that would prevail in the intestine. Just for an example, I've given this sentence. So, children from rural Africa who primarily eat more, plant-based diet, more fiber, have higher levels of actinobacteria and bacteroidetes, as against firmicutes and proteobacteria, which occur in higher abundance in Western European children who are mostly into high fat, sugar, low fiber, and protein diet. It goes to show how malleable the gut microflora can be. Uh, so non-digestible carbohydrates present in whole grains. This is some of the reports which just I picked up one of the sentences there, led to reductions in the pro-inflammatory cytokine IL-6 and insulin resistance. Again, how this gut microflora indirectly can be modulated is by something called as prebiotics. So prebiotics are stuff which will help a certain subset of good microorganisms to grow, and therefore affect certain changes. And one of the examples here is the protein extracts of B and P was shown to increase gut commensal bacteria, bifidobacterium, and lactobacillus. While V is additionally shown to decrease the pathogenic bacteria, Bacteroides fragilis and Clostridium perfringens. So there are many examples. I mean, as I said, I mean, there can be a full session on 5 hours to discuss on all these things. Suffice to say that gut microbiota gets affected by the food that we take, and therefore it's very important that we improve the reservoir of our gut microflora. Now, what happens in the elderly, the immunocompromised, and the comorbid patients? There are few facts that are known. For example, with age, the gut diversity decreases. So although it's a very hyped kind of a topic, still it is something that we cannot brush under the carpet. So when the gut diversity decreases, it definitely impacts the kind of microflora that these subset of individuals would have. Similarly, as we have seen in the in the first sentence of this slide, that diabetes and even hypertension, people with diabetes and hypertension, they have a different microflora profile. So all this, if you put all these factors together, the fact that there's a gut-lung axis, the fact that the gut microbiota has a role in immunity, the fact that diet plays a very important role, the fact that there's a decrease in diversity of gut microflora in the subset of population which are affected most by COVID-19, if you put all these pieces together, you start to see a picture, and that is where I have proposed the implications of this. Of course, more research needs to be done. So what I've suggested is the following. So you have a dysbiosis states in the vulnerables, which is the elderly, immunocompromised, and the comorbid patients like type 2 diabetes and hypertensive patients. Now, based on the gut microflora of these patients, if we can design or define functional food which will be personalized. I'm not saying one size fits all kind of an approach. So here it is very important that we actually look at the microflora of these subset individual people, because each one will have different microflora, and then define the kind of food that is required for such people and provide that as a supplement to the treatment modalities that is being provided. That might improve the gut microflora in these patients, and thereby if you see the role the gut microflora plays on immunity and the other aspects of it, then there's a chance that it will lead to better and faster recovery. Of course, this needs to be tested in a trial, and that's what I'm proposing in the paper. The second thing is something which is more dramatic, which is a role of these things as a prophylaxis or as a prophylactic agent. So people who are elderly people, if they can be given, even if they are not infected right now, if they can be given functional food based on their gut microbiota profile, so as to again improve it before the infection strikes them. So there's a chance that it will improve the immunity and improve the resistance to the infection. So in the prophylaxis, I've also mentioned in the paper that even the frontline warriors, like the doctors, the nurses, the technicians, the police people, even their gut microflora can be checked, and maybe all these supplements can be given to them in a personalized manner to see if that can improve their resistance to the infection. So with that, I end the presentation, and Dr. Kar, it's up to you to ask any questions.
Thank you, Debojyoti. That was excellent. So, going back to your personalized nutritional interventions, I just want to make a few comments in that. Number one, yes, what you rightly pointed out is we need a whole lot more research. But right now, we are still able to do some of those personalized nutritional interventions. So one of the things I want to talk about is the personalized blood sugar response. So what happens is, you mentioned whole grains in that paper, and in my world, we have a lot of issues with whole grains because of the glycemic response, and particularly in people of South Asian ancestry, people from India, Pakistan, Bangladesh, these countries, Sri Lanka. So South Asians have the propensity to be severely insulin resistant, pre-diabetic. A lot of them have diabetes and don't know. And we know with the COVID-19 patients, those who have had diabetes, they've done much worse. So when I talk about personalized blood sugar response, where the gut microbiome has a huge impact, is the same food will have different reaction in terms of blood sugars in different people. So you may be able to eat, rice, and your blood sugar doesn't go up so much. However, I may eat, a roti, and I have done on myself, so I know it. So I may have some grains, whole grains, and my blood sugar is like in the diabetic range. I'm not diabetic. So this is where one level of personalization we are already able to do. So that's why it's very difficult for me and similar functional medicine doctors to give what you said, generic recommendations for food.
Correct.
So,
Correct.
but at the same time, there are some things we can still say, like for instance, eat real food. So these are going to be my generic recommendations. Eat real food. Don't use vegetable oils, highly inflammatory. Sugar is not your friend. So these are the few things. Eat food that, as Michael Pollan says, as your grandparents would recognize as food. Eat food that a third grader can pronounce. Like some of the stuff you see in the packaged food today is like, forget about third grade, even I cannot pronounce some of them. And the other thing is, of course, trans fats are really bad. So going with that, yes, these are very generic recommendations. And yesterday, I think there was a discussion on LinkedIn about food and lung disease. So I just commented the lower carbohydrate food has shown some, again, not big studies. Most of the nutrition studies are never very big, but lower carbohydrate food, number one, would also help in managing blood sugars better. And the other thing is hyperinsulinemia as well. So high insulin levels is a very pro-inflammatory state. That is again another factor. And the other thing is, Devajiti, can you just go to that slide, your final slide, where you showed all the different interventions? Just a few comments on those.
Yeah.
Yeah, this one. This is really a great one. So one is, we talked about personalized nutritional interventions, which again, you cannot do without testing. So gut microbiome would be a good one. And of course, your personalized blood sugar response, you can wear a continuous blood glucose monitor and monitor food, steps, sleep, everything. It's not just food that impacts blood sugar levels, number one. Number two, you mentioned, prebiotics, probiotics, and synbiotics. Just one, in the interest of time, I'm not going to go, I think we'll have to do a separate one on probiotics alone. So probiotics, yes, they are definitely very beneficial. Now yogurt, the yogurt that's available in the market does not contain probiotics unless the manufacturer has enriched it with probiotics, number one. Number two, probiotics, to be called as something that benefits in terms of health, has to have a colony forming unit of 10 to the power 9 billion, billion with a B, BCFU. And a lot of the probiotics, I know, particularly the ones available in India, I know that because my patients will keep sending me the pictures, they do not contain adequate quantity, number one. The number two factor, which Devajiti has been talking about in the last two videos and today as well, is the variety of the microbes species that are present. So if a probiotic contains just two, phyla, it's not really going to do anything. Don't waste your money on that. Much rather invest it on good quality food. So probiotics, to be helpful, have to be, number one, the quality has to be very good. And another important factor is, this is where, again, a lot of these things are sold without the quality control in place. The other big factor is their stability over time. So if it's a good company making it, it will be the same content at the end of its shelf life. Let's say that's 1 year down the line, versus some of them will deteriorate when they are kept in outside temperature and all those things. And the other factor that is important in terms of probiotics is you have to have it every day because it goes out with the poop. So it's not like probiotics, you just take it for 3 months and then you expect the residual effect to stay on. So those were my few comments on, this
Yes.
is really a fantastic slide. I think I'm going to use it on my presentations.
So I will add few points on the probiotics since we are on the topic. As you mentioned, I mean, the colony forming units is definitely one of the very important factors that needs to be taken in mind before consuming anything. Second is, there is something called as colonization potential. Now, and that also depends on your genetics, by the way. So
Yeah.
there are, like, I mean, if you have variants in lactase gene, for example, uh, dictates whether bifidobacterium will be able to colonize your gut or not. Okay, so that also is very important. As I said, one size does not fit all, definitely in probiotics.
Yeah.
And the third very important point here is that you might be having an enough quantity of the probiotic that you are consuming from the market. So that is not going to help you anyway, because your body, because see, all these probiotics are basically uh, are supposed to be there in your intestine. Okay, it's not like you are getting a probiotic which was supposed to be in the lungs and you are consuming it and it is going in the intestine. No, their niche is the intestine itself. Okay, so many people do not know or understand that they are consuming a probiotic which is already there in the system. So there is not going to be any help whatsoever. So these are the few things that we have to keep in mind, especially for probiotics. It is not one size fit all, and that's why this personalized testing becomes very crucial. Right now, it is still in the sci-fi stage, but I think in years to come, this will become the norm.
So thank you, Devajyoti, for bringing that up. That was going to be my question, which was more like a big picture thinking. So again, this is going by whatever I have been reading. My vision is there will come a time when we'll be able to look at the gut microbiome and recommend personalized probiotics. So there will be companies who will be able to manufacture that. Now, I know a lot of things have to happen in between. So that dream is probably, I don't even know how many years away, because it's just not just only the gut microbiome. There is the exposome, the metabolomics, proteomics, genes,
Of course,
then of course the social network and numerous things. So my
absolutely.
question to you, Devajyoti, is if my vision and your vision as well of personalized probiotics were to come true, what are the things that need to happen from a perspective of science and all that? Now, first and foremost, we are not there yet. We will be there. So what are the things that need to happen from a research perspective, Devajyoti, that would make that a reality?
So yeah, so Shamlam, we are already there, kind of, in the sense our report provides a very important section, which is the probiotics section. We actually tell our patients that these are the probiotics, and these are GRAS. I mean, generally recognized as safe probiotics. So we are not looking at all probiotics, but only
Yeah.
GRAS probiotics, and we tell our clients or the patients that these are the probiotics that are missing in your intestine, which is supposed to be at this level, which is not in this level. And we also provide the natural sources of those probiotics. But as you said, I mean, there are challenges in the probiotic personalized probiotic space, because uh, for an individual to have a personalized probiotic, the probiotic needs to be there. Not all probiotics are there. That is number one. Second, not all probiotics can be provided in a similar form. So some of the probiotics can be provided in a powder forms because it can be lyophilized. Some of them can be only provided in a liquid form. They need to be refrigerated. Some of them they can be kept at room temperature, so on and so forth. So a lot of research at that level needs to be done to make things, it's like a tailor-made medicine or tailor-made supplement. Unless the research at the microbiology level, where you have fermentation and all those technologies parallelly, ramping up their R&D and coming up with a similar normalized way of handling probiotics, uh, the personalized probiotic space will be some years down the line, but it is not there right now. Uh, one interesting point that we have seen from our studies is that uh, there are, not all probiotics are less. Okay, in a population. Let's say we mostly deal with Indian samples, so we know that. It's only that we found that 3, 4, 5, maximum 7 or 8 probiotics that consistently show a decreased trend. That means if we can address that, I mean, we need more samples. We need the N has to be really high. That means if we can focus on those probiotics, then I think we will be reaching that dream of giving personalized probiotics much faster. So that is what I would say right now on this.
Thank you, Devajit. So what, if I'm understanding correctly, what you're saying is one is from the testing perspective.
Yeah.
So obviously we need a lot more Indian data. So that is where your company would come into the picture. So people can, at least if not anything else, they can contribute the data to the database, right? And India is so diverse. We have so many different types of food we eat. Some people are vegetarians from birth. Some of us are non-vegetarians. Some people who the indigenous population still eat a whole lot less of processed food. But I think that's changing now because processed food is so easily available.
Yeah.
So
True.
those things. And the second thing that if I'm right in understanding what you're saying is in the world of making probiotics, that is an
Right.
entire industry which needs a lot of,
Correct.
and what I understand, it needs a lot of investment because it's not easy
Absolutely.
to make high quality probiotics. We have access in Canada, we have access to some very good quality products, but in India, I have not yet found that high level. Like I don't find anything which is like 100 billion colony forming units. And some of the disease conditions that you deal with, things like inflammatory bowel disease, for example, these need much higher dose of probiotics. So rather than people saying, oh, probiotics didn't do anything to me, it's very likely that you didn't get the right one. So
Correct.
that is
Correct.
the other
Absolutely.
thing. And
Absolutely.
another thing, Devajyoti, is the genetic interaction between all of this, because in my world, food is signals to the genes, and the gut microbiome, all the genetic information from that. How much of this would need a lot of Indian data? Like we know that there is a lot of data from Western population and all that. So, and how far are we in that? And again, what else needs to happen? Because India is like 1.3 billion people. So for us to have a sample size which is significant enough, I guess that is going to be a lot, right?
Yeah. So there are two things, Dr. Kerr. One is definitely, as you mentioned, you touched upon this point of genetics, role of food affecting the genes and so on and so forth. Just to give you an example, uh, in the microbiota field, there is something called as SCFA,
Mm.
short chain fatty acids,
Fatty acids, yeah.
like propionate, butyrate, acetate, and so on and so forth. Now, butyrate is known to be an HDAC inhibitor.
Mm-hmm.
Okay, which is basically what I'm trying to say is it can affect the epigenetics of the thing. So yes, see, when we are looking at microbiota, we generally think of them as bacteria or fungi or viruses. What we somehow skip is the metabolites that these guys
Yes.
secrete.
Yeah.
And it is not just the bacteria or the virus which is doing the trick. It is the metabolites also, if not the primary thing which is affecting whatever we are seeing as a in the host. So 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 definitely these things need to be looked into in much greater depth. People have done it, people are doing it, and with more awareness coming in the field of the gut microbiome, uh, people will even more do it now. So that is I think the first part of your point, the genetics part. The second part was, I think, as you rightly pointed, more research on how to culture the probiotics, increase their shelf life, improve their colonization potential. Uh, those things need to be done. And coming to the data, see, India is a heterogeneous country. India is, a lot of European countries are just part of, let's say, the India. India will encompass a lot of European countries, You move from Karnataka to Andhra, you see a different pattern of food. And as we have been discussing, food plays a very important role in the microflora of individuals. So, India therefore gives a very, very interesting source of information to do research on. Ultimately, the success of microbiome field will depend on the kind of research that comes out. And if we rely too much on the Western research, we will be missing a lot of these points.
Yeah.
So India therefore provides a very, very good platform to researchers, to investors, to invest in companies which are in this domain, and help progress the understanding of life science better. So that is true. I mean, India provides that opportunity to, unlike other fields maybe, but gut microbiota, because of its diversity, India
Yes.
definitely provides a very good platform.
Thank you, Debojyoti. So just one more comment on, so what are synbiotics? Synbiotics are combinations of prebiotics and probiotics together. And one short comment on, fermented food. So what happens is nowadays a lot of people are, they are into taking kimchi and sauerkraut and all these things. And I have patients asking me, should I have it every day? So I have to warn them. See, the thing is, most Indians are not traditionally sauerkraut kimchi eating population. And as we know from a lot of research papers, and I think quite a few have come from Japan as well. So this was about the soy processing enzymes in the Japanese gut versus the soy processing enzymes in American gut. So it is different. My point is, just because kimchi is a fermented food, it may not be completely right for you. So again, we don't have enough data, so at this point in time, don't overdose. That is what I want to suggest. And another thing is, chocolate is fermented food, only the problem is with the sugar in it. So thank you very much, Devajyoti, and we look forward to a lot more discussions. You mentioned lactase, and I think we're going to do one on on the lactose intolerance. That's a very interesting concept. And thank you to all of you. And please post your questions either on LinkedIn or on the YouTube channel as well. Thank you very much. Thank you, Devajyoti.
Thank you, Dr. Kar.